A-Level Biology 9700, Paper 5, Variant 2, May-June 2025. And in this video, we will discuss the question number 1. And question number 1 is on screen. You can see phosphatase are enzymes that catalyze the removal of phosphate from other molecules, releasing inorganic phosphate. Phosphatase can be extracted from mung bean seedlings, Vigna radiata, and figure 1.1 shows the mung bean seedlings. Mung bean seedlings have been shown in figure 1.1. Now, a student found a published method to extract phosphatase from mung bean seedlings. Use a pestle and mortar to grind mung bean seedlings with a small volume of distilled water to make a paste. Add distilled water to paste to make a mixture with a total volume of 50 cubic centimeter. Then filter the mixture and put the filtrate into a clean centrifuge machine or tube. Centrifuge the uh, filtrate until a solid pellet is formed as shown in figure 1.2. Pour the liquid extract containing phosphatase into a clean test tube. So you can see a plastic centrifuge tube and liquid phosphatase extract and solid pellet is at the base. Now, question is, state two other variables that should be standardized in the published method so that extracts with the same concentration of phosphatase can be produced. So here we have to write two variables. The most appropriate variables to standardize for this specific method can be number one, the mass of mung bean seedlings. So why the mass of the mung bean seedlings should be the same to ensure the same starting amount of biological tissue. And second, the age of seedlings to ensure the seedlings are at the same metabolic stage. So the grinding time should also be the same. So to ensure a consistent uh, degree of cell lysis, or you can say the breakdown. So in this way, or the size of those mung bean which are produced as a result of this lysis. So I would like to write only two of them. They are number one, the mass should be same to ensure the same starting amount of biological tissue. And the second, same age seedlings should be used to ensure that all are at the same metabolic stage. Now the next question is B part, which is The student read that the optimum pH for phosphatase extracted from mung bean seedlings is less than pH 7. Less than pH 7. Less than pH 7 means it is moving towards the acidity. So 7 is neutral. Less than 7 means it should be less than alkaline. So the student decided to investigate the phosphatase activity using the substrate phenolphthalein phosphate. So in the first four steps of the method, the student mixed 5 cubic centimeter of pH 6 buffer solution for 1 cubic centimeter of a 1% solution of phenolphthalein phosphate in a test tube. Add 1 cubic centimeter of the phosphatase extract to the test tube and started a timer. Uh, Incubated the test tube for 10 minutes in a water bath at 30 degrees centigrade. The temperature. So stop the enzyme reaction after 10 minutes by adding 5 cubic centimeter of 10% solution of the sodium carbonate. Sodium carbonate solution is alkaline. It means it is more than 7. Now, the buffered phenolphthalein phosphate solution and the phosphatase extract were mixed and then placed in the water bath. The student identified this as a source of error in the method. So explain why this is source of error and state how you would modify the method to remove this source of error. So first of all, we have to write down why it is source of error, then how can we modify it? How can we modify this? So in this uh, in this method, the primary issue is that the temperature is not controlled at the exact moment the reaction begins. So what happened when you mix the buffered phenolphthalein phosphate solution and the phosphatase extract at room temperature and then place them in a 30 degree centigrade water bath, the mixture takes time to reach the target temperature because both of these solutions have different temperatures at that time. When you mix the buffer solution, it is less than 30 degrees centigrade or both are, we can say that it was at room temperature. So during those first few minutes of the 10-minute incubation, the enzyme is working at a lower changing temperature rather than a constant 30 degrees centigrade. Because when we will mix the buffer solution into it, its temperature will become less than 30 degrees centigrade. So because of the different tubes might warm up at slightly different rates, the results won't be truly representative of the enzyme's activity as 30 degrees centigrade, making the data less valid and harder to compare. So for this purpose, we should write the answer, that is that is temperature is not controlled at the exact moment the reaction takes. buffer solution and phosphatase enzyme at at room temperature. Water bath. The mixture takes time. Takes time to reach the target temperature. So it can cause the errors in the experiment because of the difference in the temperature. So then now we have to modify it. We can fix this. How can we fix this? For this purpose, we must ensure both the enzymes and the substrate are already at the target temperature before they ever touch each other. So I would write and then and or we can place the test tubes containing the buffered phenolphthalein phosphate solution and a separate tube containing the phosphatase extract into the 30-degree water bath before mixing. So both of they should be at 30 degrees centigrade before mixing. To fix this, you must ensure both the enzyme extract and the substrate are already at the target temperature. Before mixing, or or set their temperatures separately at 30 degrees centigrade before mixing to avoid error. Now the next question is, suggest how the addition of 10% solution of sodium carbonate stops the enzyme reaction. Sodium carbonate, 10% solution of sodium carbonate stop the enzyme reaction. The addition of a 10% sodium carbonate solution will stop reaction by changing the environment of the enzyme because it will change the pH. Sodium carbonate, is an alkaline solution. When we add it into it, it will significantly raises the pH of the reaction mixture. And you can see the reaction will take place on less than pH 7. So sudden shift to an alkaline pH will denature the enzyme. And it should be less than 7. So it is above 7. And the next question is the phosphatase catalase the removal of inorganic phosphate from phenolphthalein phosphate as shown by phenolphthalein phosphate, and phosphatase enzyme is there. It will break down the phenolphthalein phosphate into phenolphthalein and inorganic phosphate. So the 10% solution of sodium carbonate added at the end of the experiment also causes any phenolphthalein to turn pink. The intensity of the pink color is an indication of the concentration of phenolphthalein. To estimate the concentration of phenolphthalein produced by the reaction, the student decided to make a proportional dilution using a 2% stock solution of phenolphthalein. The student made 50 cubic centimeter of each diluted solution. Describe a method the student could use to make a proportional dilution of the 2% stock solution. So here we have to write down the method. How can we make the different concentrations? So for this purpose, I would uh, describe the method. Uh, to create a proportional dilution for a range of naphthalene concentrations, we know that the stock solution is 2%. And we have to mix varying volumes of distilled water into it to make the different concentrations of the solution. For this purpose, uh, we will make the use of the formula, which is C1V1 is equal to C2V2, where C1 is the stock concentration, which is 2.0% already given to us. V2 is the final volume, which is 50 cubic centimeter. We have to make this volume of each concentration. And then C2 is desired concentration, which we will make over here as, for example, 1.5, 1.0, 0.5. Okay, we have to make between 0 to 2%. So all different concentrations can be included into it. It depends on you which are you are making. Here I am going with the and the V2, the volume of water added. So here if we make the table for this purpose, which is let me We have to make 5 concentrations over here. So first of all, as they have given the desired concentration, which is C2, and then volume of stock solution, and then which is V1. This is V1, C2 V1, and then V2, volume of water, distilled water, and then total, and then volume of solvent, total volume, which is V2. Here I am going to write the so first of all, desired concentration, we will make 2%, 1.5%, then 1%, 0.5%, or 0.1%. So these 5 concentrations. For this purpose, you can make use of this formula to calculate this. I have calculated. So it is 50 and 0, and total will be volume will, and it will be equal to 50. And when we have 1.5, it is volume of the solvent 37.5. So it will be equal to 12.5, and both of them will make 50. And then when there is 1%, then it will 25, and 25 water will be added into it. So it will become equal to 50. And when there is 0.5% concentration, it depends on you. You can choose any of the concentrations. Beside this, you can choose. It depends on you. 37.5, again 50, and then 2.5, and it will be equal to 47.5. Again, it will be equal to 50. So it means, so how do we have do this? By using this equation, with this formula, by using this formula, we can calculate. So you can calculate with the help of this formula. Now the next question is, describe how the student could choose the dilutions from C and a colorimeter to estimate the concentration of phenolphthalein in a reaction mixture? Okay, the question is, actually this is a common laboratory technique which is used to calibration curve. So here I would like to discuss that first of all, uh, you must establish a relationship between a non-concentrations and their light absorbance. So as first of all, we have to use the calorimeter to measure the absorbance of each of the non-dilutions prepared in the part C, which we have already done above. So, and then we have to test the unknown mixture. Once the graph is ready, you can find the value for your reaction mixture. Then we will place the reaction mixture into the calorimeter and record its absorbance value. And then we will identify the concentration, and then we will make use of these values, and then uh, to find the to estimate the concentration of naphthalene in a reaction mixture. So I'm going to write all these points which I have discussed with you. Yes. Uh, First step is establish our relationship between known concentrations and their light absorption. Secondly, the calorimeter is used. Use calorimeter to measure the absorbance of each of the known dilution which we have done in part C. Then we have to ensure that calorimeter is set to a specific wavelength, to get the most accurate, to get the readings. Most, you can say, uh, readings. So plot a graph of absorbance against the known concentration. concentrations of phenol. Once the graph is ready, you can estimate, you can find or estimate value of reaction mixture. Now, moving towards the next question. Which is part E. The student decided to determine the optimum pH for phosphatase extracted from the mung bean seedlings. Describe a method the student would use to determine the optimum pH for phosphatase. Your method should be set out in a logical order and be detailed enough to allow another person to follow it. Details of how to extract phosphatase from the mung bean seedlings and how to make the dilutions of naphthalene solution should not be included. So we have to write the complete answer. Experimental method. So for this, we will first of all, we have to follow these steps as first of all, set up the independent variable, which is pH. So then we have to standardize the reaction mixture. Then the measurement of the dependent variable. Then we will move towards the control variables and data analysis. So these we will discuss all these points in detail in this question. So we will start with the setup. First of all, we will set up the independent variable, which is pH. So prepare a range of at least 5 different pH values. For example, 3, 5, 7, 9, or 11. And for this purpose, we can use the buffer solutions by using buffer solutions. And buffer solutions are very important to use. Then add a fixed volume of the substrate into, and substrate, phenolphthalein diphosphate into the separate test tube. For each pH. Place all tubes in a water bath at a constant temperature. For example, 30 degrees centigrade. Then mix the enzyme and substrate. Enzyme and substrate and start stopwatch. For a fixed period of time. For example, 5 or 10 minutes. Stop the reaction by adding alkali use a colorimeter to measure the absorbance of each sample. More or higher absorbance will indicate more phenolphthalein was released. Temperature should be controlled controlled water baths should be used. Plot a graph of rate of reaction against pH. repeat the experiment. With a narrower range of keys to avoid or to get accuracy. So it was all about part E. Moving towards the next question. And next question is, the student investigated the effect of competitive inhibitor on the activity of a phosphatase. Figure 1.3 shows a graph of the initial rates of enzyme reaction against phenolphthalein phosphate concentration for the enzyme with no inhibitor and the enzyme with inhibitor. So here you can see the one of them, the graph. So with dots, it is enzyme with inhibitor, and other one is with a cross with no inhibitor. So what is question? One of the data plots in figure 1.3 is anomalous. Circle the anomalous data plot in figure 1.3 and explain why you think it is anomalous. So moving towards the graphs, or in graphs, you can see the one of them value is anomalous. So we will circle this. This value will be circled. And why we have circled? Because you can see it is above the V maximum. So it is above the V maximum. So this value is anomalous. I think we should write over here instead of typing the given space, or we will move towards that. Yes. This is above the V maximum, which is not a correct value. Moving towards the next question. Next question is, use figure 1.3. Determine the Michael-Menten constant for the enzyme with no inhibitor and for the enzyme with inhibitor. Include the correct units in your answer. So the V maximum for both of them are, we will moving towards the graph with inhibitor and with no inhibitor. So, to find out the Michael-Menten constant, first of all, we have to go with the half of the V maximum. So half of the the maximum is 0. This is 0.08 for both of them. with inhibitor or without inhibitor. So first of all, and half of them is, so it is half. So this is the half value. So we will go with this, and when we move in the downward direction, please make use of a scale. I am doing without scale. Yes, it is not possible for me on laptop to use the scale. It is less than 0.5. Yes, it is less than 0.5. So, and this point, and this point, this is this point, and this point. So the one value is 0.75, and other value is so with inhibitor is with no inhibitor is 0 point and with inhibitor is 0.425. So these are the two values. Now question is, calculate the percentage increase. And you know what is the formula for percentage increase is 0.1. So here percentage increase formula is equal to new value minus initial value over value multiplied by 100. So here the new value is 0.425 minus 0.075 divided by 0.075 multiplied by 100. So in this way you can find out the answer with this formula. So you can calculate with the help of calculator. So it was all about question number 1. Please like, share and subscribe my channel. Thanks for watching.